Daniel Luksic, and today I'd like to talk about an article that was recently published in Kidney 360 called Clear Evidence of LAMA5 Gene Biallelic Truncating Variants Causing Infantile Nephrotic Syndrome. I think this article is interesting for a number of reasons, such as their use of molecular methods to confirm the functional changes in the genetic mutations they found, and it also details a good example of the genotype-phenotype correlation seen in this gene. In about 10 to 20% of the cases of nephrotic syndrome in pediatric patients, the condition is said to be steroid resistant. And of these steroid resistant nephrotic syndrome cases investigated in recent studies, about a quarter of them have had an identifiable genetic cause. Now, it has been observed that some pediatric patients with steroid resistant nephrotic syndrome have had genetic findings in the LAMA5 gene, but these variants haven't been definitively confirmed to be pathogenic. LAMA5 is found in the glomerular basement membrane, which is made up of four main types of proteins, including nitrogen, heparan sulfate, proteoglycans, laminin, and the collagen 4 proteins, which you may know as the proteins which are mutated in Alport syndrome. In the glomerular basement membrane, laminin is most often found as laminin 521, which is a heterotrimer of the alpha 5, beta 2, and gamma 1 subunits. LAMB2, the gene responsible for the beta 2 subunit, is already known to be associated with nephrotic syndrome. The current study looked at 407 Japanese patients who were identified after presenting with severe proteinuria. Next-generation sequencing was done with a panel of 128 genes on these patients, as well as some further genetic analysis of RNA isolated from cells in the urine. Additional analysis of genetic variants found in these patients was done by creating cell lines which expressed copies of the mutant LAMA5 genes, and histology was performed on these cell lines to assess the resulting protein function. Of the 407 patients, 4 individuals from 3 families were identified with biallelic mutations in LAMA5. This paper provides clinical details on the patients and their genetics, which I will summarize here. Patients 1 and 2 were siblings who presented at 3 and 4 months old with nephrotic syndrome and were found to have biallelic truncating mutations in LAMA5. Patient 3 was also found to have nephrotic syndrome, this time at 6 months old, and also had biallelic truncating mutations. Finally, patient 4, who didn't present until 8 years old with proteinuria, was found to have biallelic missense mutations. Missense mutations often correlate with less severe changes to protein function and often less severe disease progression as well. Cell lines were then made to express the LAMA5 gene mutations that each patient carried, and the resulting immunostaining revealed that each genotype had reduced amounts of the laminin 521 protein being expressed. In this study, patients with biallelic mutations in LAMA5 were seen to have decreased protein production on histology and a corresponding disease of the glomerular basement membrane. These current patients all had a clinical heterogeneity, which to a point corresponds to their genotypes. Those with more severe mutations, i.e. the truncating mutations, led to more severe presentation and an earlier age of onset than those with the missense mutations. This type of phylogenetic heterogeneity is also seen in LAMB2 mutation carriers. LAMB2, which forms another part of the laminin 521 protein complex, is also associated with a nephrotic syndrome. These functional studies shed light on the genotype-phenotype correlation that is seen in this gene. The alpha 5 subunit of the laminin 521 protein complex has a cross-like structure containing a beginning section that coils around the other 2 subunits and a later section which binds to the other elements of the glomerular basement membrane. In mutants that are seen earlier in the protein, that is, where the laminin A5 binds to the other 2 subunits, that heterotrimer isn't formed at all, and the mutations are known in these cases to be lethal and to cause multisystemic presentations. In the current paper, all of the mutations that were identified were seen thousands of base pairs later, after the location of these lethal mutations, in the part of the protein not involved in binding to the other subunits. This is why the laminin in these patients was still allowed to trimerize and the patients were seen to survive. As the authors say, quote, we think it's likely that patients with nephrotic syndrome with these types of LAMA5 variants are so rare because only specific types of LAMA5 variants are compatible with life. And even in those that are compatible with life, we can still see a correlation between the genotype identified in the patient and the phenotype that they present with. The authors remark that LAMA5 is potentially a cause of infantile nephrotic syndrome if a rare one, and recommend that in the future this gene be included on genetic evaluation of infants who are presenting with nephrotic syndrome. Thank you.